for decades, doctors looked at the really severe disfiguring skin lesions of lupus patients. And, well, they basically just blamed the immune system.
Right. The systemic immune system was always the prime suspect.
Exactly. I mean, think about it. A patient with cutaneous lupus erythematosus walks out into the sunlight. You or I, we might get a mild sunburn if we forget our SPF, you know?
Yeah, maybe a little redness, a little peeling.
But for someone with CLA, that single ray of sunlight triggers this massive, cascading, chronic flare-up. And the medical consensus was always to just point a finger at the systemic immune system.
The thought process was pretty simple, really. The immune system is hyperactive, it's overreacting to the sun, and so it's attacking the skin.
So the logical response to that diagnosis has always been systemic treatment.
Yeah, heavy-duty immunosuppressants. You're basically quieting the entire body's defense mechanisms just to save the skin.
Which takes a huge toll on you as a patient. But, and this is why we're here today, a groundbreaking 2026 paper from Cells Death and Disease proves the medical establishment has been looking at the wrong culprit this entire time.
Entirely the wrong culprit. It's a total paradigm shift.
So, welcome to another deep dive. Our mission today is to tear into this new research, because the true instigators of these lupus skin flare-ups are not the systemic immune cells rushing in unprovoked.
No, they're not.
The instigators are the skin cells themselves. They're locked in this microscopic self-amplifying cycle of destruction. We're going to explore the single-cell RNA sequencing that uncovered this mystery, and then look at this wildly futuristic but remarkably elegant solution.
The dissolvable microneedle patches.
Yes, engineered to literally short-circuit the disease.
So the foundation of this entire discovery rests on rethinking the primary cells of the epidermis, which are the keratinocytes.
The keratinocyte.
Right. Historically, dermatology viewed these cells as just, well, like bricks in a wall.
Just structural.
Exactly. They were considered passive victims of UV radiation, just sitting there taking the damage while the immune system went completely haywire around them.
Let's use an analogy here to help visualize this. We've always treated the immune system like a police force and the skin like a highly secure building.
I like that.
And the old assumption was that the police were just, randomly showing up and attacking the building. But mapping the cutaneous architecture of lupus-affected skin revealed something entirely different.
It turns out the security guards, the keratinocytes themselves, have actually gone rogue.
Right. They are actively starting a riot inside the building.
Right.
And purposefully calling the police in to escalate the chaos.
And the researchers proved this by using single-cell transcriptomics, which is a really powerful tool.
So they looked at 14 normal skin samples and 7 lupus skin samples, right?
Exactly. They analyzed the gene expression of every single cell. And when they isolated the keratinocytes, they didn't just find one uniform type of cell.
They found 9.
9 transcriptionally distinct subclusters, yes. And 2 of those groups, subcluster 3, which is stress responsive, and subcluster 7, which is immune modulatory, those were massively expanded in the lupus patients.
So these specific rogue security guards, they carry a very distinct genetic signature, don't they?
They do. They show this massive interferon signature.
Meaning like elevated expression of genes like MX1, OAS1, and ISG15,
right,
which is a classic hallmark of lupus pathophysiology.
Right, and it was entirely absent in the healthy, normal keratinocyte subclusters.
Okay, let's unpack this. Identifying the rogue cells is only the first step. The next hurdle is figuring out what these specific subclusters are actually producing to initiate this riot.
And the initial assumption would naturally be your classic inflammatory markers, like TNF-alpha or IL-6.
The usual suspects.
The usual suspects, but they were barely present. Instead, the transcriptomics revealed these cells were pumping out a molecule called MIF.
MIF, macrophage migration inhibitory factor.
Exactly. It's a really potent driver of inflammation.
And the mRNA levels for this specific cytokine were spiked precisely within those pathogenic subclusters, 3 and 7. Even before overt visible lesions had fully formed on the skin.
Which is wild, and they verified this across the board using immunohistochemistry on clinical skin samples.
Right, for acute, subacute, and chronic CLE.
Across every major subtype, there was a massive upregulation of MIF protein in the epidermis compared to normal skin.
So just to recap this first part for you listening, because I want to make sure we lock this in. Bullet 1, UVB sunlight is the trigger. Bullet 2, keratinocytes, specifically subclusters 3 and 7, are the active culprits here. And bullet 3, this MIF molecule is the weapon they produce.
That's a perfect summary.
Have you ever thought about your own skin cells as active communicators rather than just a physical barrier protecting you from the outside world? It's I mean, it's a huge shift in perspective.
It really is. It changes how we view the skin entirely.
But hold on, though. Now that we know MIF is the weapon hiding inside these cells, exactly how does the physical stress of sunlight pull the trigger to unleash it? Because, I get sunburned all the time and my skin doesn't erupt into chronic autoimmune lesions.
Right, which is the big question. If UVB is the universal trigger, why is this catastrophic reaction localized only to lupus patients?
Right.
Well, the difference lies in how the lupus keratinocytes are primed.
Primed,
Yeah. These cells suffer from a chronic buildup of endogenous nucleic acids, or ENAs, in their cellular fluid.
So their own genetic debris.
Exactly. Because they lack the ability to efficiently clear out their own nucleic acids, they exist in this constant state of cellular paranoia. They are hypersensitive.
They're essentially living in a hoarder's house filled with their own genetic trash, making them incredibly reactive to any outside stress.
That's a great way to put it. And to prove this was the differentiator, the researchers actually simulated it.
Oh, really?
They took normal human keratinocytes and transfected them with ENAs, artificially creating a disease-relevant lupus-primed model.
Wow.
And when UVB hits these primed cells, it triggers a highly specific cascade known as the ribotoxic stress response, or RSR.
I want to trace this RSR cascade step by step because the mechanics here are just fascinating. The millisecond the UVB damages the RNA in the cell, it trips a sensor molecule called ZK-alpha.
ZK-alpha is the alarm bell. Once it's phosphorylated and activated, it triggers a downstream messenger protein called P38.
And P38 is like a major signaling hub in the cell, right?
It's a huge signaling hub. Its primary target in this specific scenario is a transcription factor called CEBP-beta.
Okay, CEBP-beta.
So P38 goes to work, and it actually does two things to CEBP-beta. It simultaneously increases the overall concentration of it and phosphorylates it to basically supercharge its activity.
So we have a chain of command here. The sunlight trips the ZK-alpha alarm, which dispatches P38, which then supercharges CEBP-beta. What does CEBP-beta actually do once it's supercharged?
It acts like a factory boss. It travels directly into the nucleus of the cell. And the researchers used a chink IP assay to prove that it physically binds to very specific regions, fragments 1 and 2 of the promoter for a gene called NLRP3.
And it commands the cell to massively upregulate NLRP3 production.
Exactly. And this specific routing to NLRP3 is a severe deviation from healthy biology.
Because normal cells don't do this.
Right. In normal unprimed keratinocytes, UVB exposure primarily activates a different molecule altogether, NLRP1.
Oh, NLRP1.
But the lupus-primed environment forces a complete switch. The stress signal bypasses the normal NLRP1 pathway and routes entirely through this P38 to CEBP-beta pathway to crank out NLRP1.
Okay, another quick visual memory tool, just a mini recap.
Yeah.
Imagine a falling row of dominoes, right? UVB knocks over ZK-alpha, which knocks over P38, which activates CEBP-beta, which finally turns on NLRP3.
The domino effect is perfectly described there.
Oh, wait, we established earlier that MIF is the ultimate weapon these cells deploy, and the cell is manufacturing all this NLRP3 internally.
Right.
So how does the inflammatory MIF protein actually break out of the cell to cause damage in the surrounding tissue?
Well, biology usually relies on, highly regulated transport systems.
Right, like vesicles.
Exactly. Cells politely package proteins into tiny microscopic delivery boxes, like vesicles, and ship them out through the Golgi apparatus or via exosomes.
So I assume the researchers chemically blockaded the cell's standard shipping routes to see if the MIF would get trapped inside.
They did. They used inhibitors for these pathways like Berfeldin A and GW4869. They shut down all the polite pathways. The MIF didn't even slow down. It kept pouring out into the surrounding environment, proving the cell was using an entirely different, non-traditional exit strategy.
Here's where it gets really interesting. If the cell isn't boxing up the MIF and shipping it out, the alternative is brutal. The exit strategy is actually a highly violent form of cell death called pyroptosis.
The term literally translates to a fiery death. That newly minted NLRP3 machinery we talked about, it acts like a microscopic pair of scissors. It seeks out a specific protein inside the cell called GSDMD and cleaves it in half.
It basically decapitates the protein, and the severed N-terminal fragment becomes structurally dangerous.
Highly dangerous. It travels to the cell membrane and physically inserts itself, punching massive hollow pores right through the cell wall.
It's like popping a water balloon with needles, right?
Exactly like that. Instead of the cell bursting completely all at once, dozens of these large GSDMD hollow needles embed themselves into the membrane, and the water, or in this case the MIF, aggressively leaks out everywhere.
instead of the cell politely packaging the water into Tupperware containers.
Right. And they proved this was happening using a drug called disulfiram,
which specifically blocks GSDMD pores.
Exactly. Once the pores were blocked, the membrane stayed intact, and the MIF leak completely stopped.
Okay, mini recap time again. Bullet 1, normal packaging via vesicles is not used. Bullet 2, NLRP3 triggers GSDMD to punch pores in the membrane. And bullet 3, the cell undergoes explosive pyroptosis, spilling the MIF.
It's spot on.
Consider the sheer mechanical violence happening at a microscopic level when a cell undergoes pyroptosis. How does that local explosion ripple out to the rest of the skin?
That is the most crucial part of the pathology, because that spilled MIF doesn't just vanish, it acts as a potent signaling molecule in the extracellular space.
Where does it go?
It drifts over to neighboring healthy keratinocytes and fibroblasts in the skin, and it binds to a specific receptor on their surface called CD74.
Let me see if I'm tracking the full loop here. When the spilled MIF binds to the CD74 receptor on the neighboring cell, it instantly reactivates that exact same P38 pathway we just traced.
Yes. It creates a perfectly self-sustaining localized feedback loop.
Oh, wow.
The first cell explodes and releases MIF. That MIF hits the neighbor's receptor. The neighbor's P38 pathway activates, building more NLRP3, which chops up more GSDMD, punching more pores, and releasing even more MIF.
An infinite loop of autocrine and paracrine signaling that never shuts off. It just cascades across the epidermis.
And this constant localized explosion is what drives chronic lupus lesions. The loop constantly upregulates tumor necrosis factor alpha, and it forces aggressive tissue remodeling by upregulating matrix metalloproteinases.
Like MMP2 and MMP9, along with collagen.
Exactly. The physical architecture of the skin literally begins to chronically break down and abnormally rebuild itself, resulting in the disfiguring lesions patients suffer from.
So we have the entire vicious cycle mapped out, from the initial UV trigger down to the cellular dominoes to the explosive pyroptosis pores, and finally into the infinite cell-to-cell loop.
That's the mechanism.
But the ultimate question is, how do we break this vicious cycle in a way that actually helps patients?
Well, the research team tackled this using a classic lupus-prone mouse model, the MRLLPR mice. And their first approach was strictly genetic.
What did they do?
They injected AAV's viral vectors carrying short hairpin RNA specifically engineered to silence the MIF gene in the skin.
A microscopic viral hitman designed to take out the MIF factories before they could even start production.
And it proved the mechanism flawlessly. The skin lesions reduced, and all the localized inflammatory markers plummeted. The loop was successfully broken.
And it's but utilizing localized viral gene therapy injections for routine autoimmune flare-ups in human patients presents massive translational hurdles, doesn't it?
Huge hurdles. It requires viral vectors, it's incredibly expensive, and it just isn't practical for day-to-day disease management.
Right, because we know the target is the skin. But taking a traditional systemic pill floods the entire body, leading to those brutal immunosuppressive side effects we talked about at the top of the show.
Which is why we need a way to get a localized drug strictly to the keratinocytes without using a traditional hypodermic needle or a viral vector.
And that specific clinical need drove the engineering brilliance of this study. The ISO-1 loaded microneedle patch.
Yes, this is where the structural biology meets material science, and I love this part.
Go ahead, geek out on the details for us.
Okay, so the patch itself is a small 10 by 10 array of tiny needles made from polyvinyl pyrrolidone, or PVP.
EVP.
Right, it's a biocompatible polymer, and the physical dimensions are incredibly precise to target the epidermis. The needles are exactly 850 micrometers tall.
Wow, specific.
Very. And they can withstand 8 newtons of force, meaning they are structurally sound enough to penetrate the tough outer layer of the skin perfectly without fracturing or breaking.
So they penetrate the outer barrier seamlessly. What happens once they are actually embedded in the skin?
Because they are made of that specific PVP polymer, they immediately begin to interact with the body's natural interstitial fluid. Within 30 minutes, the needles completely dissolve into the skin.
The delivery mechanism itself melts away, releasing the chemical inhibitor, ISO-1, directly into the epidermal and dermal layers.
Exactly. It drops the payload precisely where those rogue keratinocytes are trapped in their pyroptotic feedback loop.
Because ISO-1 is a specific chemical inhibitor of MIF.
Right. And treating the lupus-prone mice with this dissolvable patch yielded remarkable results. It effectively disrupted the entire MIF P38 GSDMD loop.
without having to suppress the mouse's entire immune system.
Exactly. The levels of cleaved GSDMD, those dangerous pore-forming heads, dropped significantly. Inflammatory markers plummeted, and the physical skin lesions healed just as effectively as they did with the viral gene therapy.
So what does this all mean? Let's zoom out for our macro takeaway. For years, think about the historical treatment of lupus like using a massive house-wide sprinkler system to put out a small grease fire in the kitchen.
It's a very destructive approach.
Right. Yes, the water will eventually put out the fire,
Yeah.
but you are going to cause immense irreversible water damage to the foundation and every other room in the house. Those are the severe side effects of systemic immunosuppression.
Patients suffer from systemic infections, organ strain, and a host of other complications just to manage a skin lesion.
But this microneedle patch, it acts as a highly targeted fire extinguisher. It goes exactly to the kitchen, exactly to the source of the molecular fire, the localized keratinocyte loop, and snuffs it out right where it starts without flooding the rest of the body.
We are finally moving from blunt force systemic suppression to precision localized disruption.
It completely rewrites the diagnostic landscape for cutaneous lupus erythematosus.
It does, but, stepping back from just the skin, this forces us to look at the entire field of autoimmune research through a completely new lens.
How so?
Well, if local skin cells, the keratinocytes, can act as the primary self-sustaining engine for an autoimmune flare-up, we really have to question the driving forces behind other systemic autoimmune diseases.
Oh, wow. I hadn't thought of that.
Could they also be driven by local non-immune tissue cells? What other microscopic rogue security guards are hiding in our kidneys or our joints or our lungs?
Just waiting for a targeted, localized treatment to switch them off.
Exactly. How many other organs are locked in their own localized feedback loops, furiously calling in the immune system? It really challenges researchers to stop looking exclusively at the immune cells and start mapping the transcriptomics of the local tissue environment.
Because the cells we previously thought were just structural scaffolding might actually be pulling the strings. It shifts our entire understanding from a binary mentality to recognizing the complex, fiery communication happening cell by cell. The building isn't just under attack. The building is calling the shots.
Perfectly said.
Well, thank you for joining us on this deep dive. Keep questioning the biological world around you. Keep looking closer, and we'll catch you on the next one.